So let's briefly talk about microorganisms and the microbial community. Now that we've seen all of this information kind of from a high level, what are we trying to study actually? Okay, so the question is the R code used for each step of the pipeline shown available? No, not all of the code that you just saw was in R. Some of the code was written in C, some of it was scripts in Bash, some of it was in R, and some of it was in Python. So it's really a combination of different types of codes that were put together in a single workflow. And that workflow in the end produces these outputs, which are either files or interactive outputs. But the focus of the program will primarily be on how to use a lot of those different standard steps, as well as some coding in R. So the majority of the coding that we'll do will be in R. Hopefully that answers your question. So let's briefly talk about microorganisms and the microbial community. What are we trying to study, and how are we going to be able to study this community, right? Why is it new, and why is it useful? So the connection between disease and microorganisms has been established already in the 1800s. And already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacteria function in isolation. However, it was soon apparent that most microorganisms don't live as individuals, but live in complex communities. And today we know that microorganisms, as they are called microbiota, can be very different in different places because they form a stable community. And so one of the efforts of the community that studies the microbiome is to try to characterize both what is healthy, establish a baseline, and how do we measure deviation from that healthy condition. So here you can see that the number of studies has been exponentially increasing from the time that these methods were introduced. And today we have a pretty good sense of these different established communities in the human body, for example. So here you can see some typical members of the microbiota in the mouth, skin, and other parts of the body. And here you can see the number of studies of published research papers on human microbiota. Now, the majority of these studies focus exclusively on the bacteria, but the microbiome actually includes bacteria as well as other members of the community, including the virome, which, as you know, bacteriophages are important members of that bacterial community that regulate the composition, can infect bacteria, bacteria fights them off. So they do form they do influence each other, as well as archaea and other members as well. So when you think of the microbiome, it's really all of the microorganisms, but when we will study the types of analyses that we will perform, we'll focus on one of them. And typically it's going to be the bacteria. Now, in different parts of the body, there are different numbers of bacterial species. And in many cases, you will find an association between the environment that they live in and the community that they form. For example, acidity levels, access to oxygen, and things like that are important factors that actually determine how this community evolves, and is one of the reasons why we cannot just isolate a lot of these individuals to study them in a lab, because we would have to recreate the exact conditions where they grow. And those conditions are in part dependent on the composition of the microbiome. So it's very difficult to extract the microbiome and actually reproduce it in a lab, even though some attempts are being made. So typically, when we think of the microbiome, we think of the gut and the digestion part of how the microbiome functions, but many other communities are also as important. For example, we can talk about the skin, right? So on the skin, surprisingly, even though we wash our skin all the time, we apply different things to our skin, these bacteria form a very stable community. And the reason is because, of course, there are pores, and the bacteria populate the pores as well as live in different cracks between the skin cells. And so as a result, we can see that bacteria, fungi, and viruses form a stable community that starts interacting with the epithelium in our skin. And that is why some of these studies focused on the skin microbiome are are very important for understanding of how to treat diseases and how potentially these bacterial communities could be interrupted. So to study a lot of this, many different international projects have been started, and those include the NIH Human Microbiome Project, the British Gut Project, the American Gut Project, so different kinds of gut projects, Micro Rosetta Initiative. There's even a project for studying the microbiome in space through the NASA consortium called GeneLab. As well as different types of clinical studies involving the microbiome, like the gut microbiome sub-study for cancer prevention, or Alzheimer's gut microbiome project, or the nurses' health study. So a lot of interesting data sets have been reposited in the public domain as a result of these consortia. And we will go through some of these examples in this program, using them as case studies to really understand what kinds of questions are people asking and what kinds of information can we obtain, for example, about the individuals that are being studied, the microbiome effects on human health, as well as many other applications of the microbiome. So let's go now and talk a little bit more about where this data comes from. So we talked about the microbiome. We talked about where we want to get to. How is this data generated? So the way this data is generated is using what's called metagenomic sequencing. And so to understand the process, we'll kind of go through the history of metagenomic sequences and exactly what is being sequenced. And then that will explain a lot of the things in the metagenomic sequence analysis and our ability to annotate and interpret the results that we just saw. So we'll talk a lot here about what's called 16S amplicon sequencing. And it's a technique that was developed because the 16S ribosomal RNA gene is an established gene that has a common function across many different types of bacterial species. And the variation has specific indications for the levels of taxonomy. So the 16S ribosomal RNA sequencing is also low cost, flexible, and it provides us a long enough read that can actually answer a lot of the questions that we have about the microbiome composition. Why is that? The reason is because here you can see the 16S ribosomal RNA gene. So it folds into the structure and has what are called different regions. So these V1 to V9 are the different regions. And the full gene is approximately 1,500 base pairs long, but it also has specific conserved regions. These regions that we see poking out are conserved, and those are between 30 and 100 base pairs long. Now, in between, you have variable regions, and then you have conserved regions. And so the databases that we will use to annotate where these reads came from actually contain those sequences already annotated by the strains of bacteria that they come from. One of those databases is called the Silva database. We'll use that extensively, but also there are others like Green Genes, RDP, and the NCBI National Center for Biotechnology Information also contains that as well. Now, there have been publications that have compared how this is, how these databases annotate the different taxa using the 16S ribosomal RNA gene sequence. And it comes out that two major things are important. Number one, when everything is driven by users, there's a lot of noise because maybe there are some opposite views on which taxa that came from, but also because someone needs to validate and clean up and constantly kind of interact with the data to make sure that it's up to date. And it turns out that Silva is one of those repositories that is most actively supported, reviewed, and validated. And so that's why in a lot of cases, we'll see people use the Silva database to annotate their metagenomic data. Now, how is this taxonomic classification actually performed? So we have here the ribosomal RNA. So the ribosome consists of two parts, the larger part and the smaller, and they are called the subunits. So we have the 30S ribosomal subunit, and the gene that encodes for that subunit is the 16S ribosomal RNA gene. That gene could be amplified. And to amplify, so we extract the DNA from the microbiome, from the actual biological sample. And then we need to amplify some target region because we cannot sequence the full 1500 base pair long sequence, not with Illumina sequencers at least, but other sequencers could do that. But with Illumina sequencing, which is the most reliable and cost-effective in this case, you can see that the primer sequence has to be designed for specific target regions. Now, different combinations of these hypervariable regions are used for the actual sequencing. Sometimes it's just V3, sometimes just V4, sometimes V3 and V4 together. Sometimes you can add another one of these hypervariable regions. It really depends on the level of taxonomy that you would like to use and the ability to amplify and then sequence those reads. What essentially happens is that with this amplicon approach, if the less you amplify, the less of your data is going to be used. So the less information will you be able to expect from your data. A comparison between the full 16S ribosomal RNA gene, for example, sequenced with Nanopore or with PacBio, compared to Illumina, which would only use one of the hypervariable regions, is that you lose about 50% of your data. So you get 50% more information if you sequence the full gene. Obviously, variation in combinations of the hypervariable regions also play a role. But what you can do as a result is actually look at the different levels, right? So the more hypervariable regions you have, the deeper you can go in the taxonomy. So instead of just looking at the species, which obviously bacteria is just one, kingdom, instead of going through the kingdom, bacteria is just the kingdom, you can go all the way to the species if you sequence everything. If you only sequence some, for example, 2, maybe you can only go to order or family level and not any deeper. But essentially then you can look at the body, for example, and analyze the composition, right? So proportions of different types of taxonomies and compare them between individuals, between groups of individuals, or find associations between individuals or microbiota and some phenotype. So this will be the data preparation, the biological data preparation, and this would be the analysis. Now, this is just based on the sequence data. What other types of data can be used for understanding not only what the composition is, but also, importantly, we want to understand the function. So for sequencing, DNA-based approaches like 16S ribosomal RNA or ITS gene sequencing could be used. ITS is just another gene that is used for assigning taxonomy. But also we can use metatranscriptomics or full metagenomics. So full metagenomics right here means that we're trying to not only select a gene for taxonomy, but we would like to then assemble everything that we sequence into full genomes. That's a challenge because we don't know all of the genomes. So there's not a comprehensive database to say, these are all the possible genomes, let's just find those. We're always expecting to find new genomes. And because they live in a community, we expect to be some drift, genetic drift, a recombination, and other types of interactions that we really cannot predict. And so that makes whole genome metagenomics challenging. Another approach is to try to understand, well, what genes are being expressed? Again, here the problem is we don't know all the genes that could be present, so how do we measure their abundance, right? How do we denoise this data? And so that is why the majority of the studies are still relying on 16S ribosomal RNA. However, more and more whole genome metagenomics is being performed, and even some metatranscriptomics is being done today as well. In addition to that, we can study the proteins and metabolites that are being produced. And that gives us another important insight into not only what is being done in the community, but how is that community interacting with the host. And to do that, ultimately, we would like to integrate all of this information together. And that brings us to systems medicine approach, or systems biology approach, to be able to understand how all of this information between the types of bacteria, the types of viruses, the types of hosts that they are in, and the different impacts of variables like diet, drugs, and other things can play a role in this complex system. So we won't go all the way into that portion of it, of how to integrate all that data, but we will definitely give you a good sense of how you can get started with metagenomics, and then hopefully that will give you enough tools to continue and study other methods as well that we will also potentially cover in other programs. So let's talk now about some practical examples and case studies that we're going to review in this program. One interesting repository is the American gut sample. So this is a project that was started, first of all, as a research study, but then was crowdfunded and really offered to anyone who would like to participate to be able to understand several different things. First of all, I would like to understand about my own gut sample. What is in it? What are the proportions, and what is the taxonomy? And maybe something is known about these specific strains or species in my body. Another important insight is that I could compare my microbiome compared to other microbiomes of other individuals. And since I know something about those individuals, for example, what kinds of diets they have or what kinds of activity they do throughout the week, I could then understand maybe where I stand in terms of the comparison. So here, this large dot represents this individual, and the little dots represent some of the other members of this project. And here the same way. So here, this project, the data is accessible online. So what we'll do is we'll actually take some of the metadata, and here you can see just a sample comparison that we will do, where we will take individuals that exercise rarely, exercise regularly, and we will try to extract their data using the sample ID and process this data to compare them to each other. And we'll ask the question, does exercise in fact have any effect on the microbiome composition? Another example project that we'll talk about is the skin microbiome. How do we study inflammation? Acne causes inflammation, of course, skin rashes. How can we actually look at the different types of inflammation caused by these microbial communities and just find associations, right? So again, what's important here is that we are dealing with noisy data. A lot of the information that we need is missing. And when we ask these questions, we ask pretty general questions. And then if we want to go beyond the general questions, then we want to actually functionally validate that what we are saying is true. A lot of times we cannot perform those types of experiments on individuals. So what we'll do is we'll look at a mouse study that actually tries to isolate the impact that the microbiome composition plays in the relationship between obesity and anxiety. Because really, today, when you look at the literature that describes microbial communities, you will see that a lot of that is potentially very impactful, but the question is, is that really true, right? So there's a lot of microbiome, theoretically could be involved in almost anything, but how do you know that it's microbiome and not anything else?